Hello everybody. Welcome to my journal club presentation. Today we're going to be going over the article I selected, which is called Gut Microbiome Strain Sharing Within Isolated Village Social Networks. So this is actually a recent article that was published in Nature in 2025, and it asks the question, how can face-to-face social interaction shape the microbiome? So specifically, the researchers wanted to look into different types of relationships, so not only people living in the same household, but also close friends. And they examined how these individuals are spending time together and how often. And they did this by sequencing the gut microbiome of 1,787 adults from isolated villages in Honduras. And they also mapped social networks in the villages by surveying the individuals that had given samples. So both of these data sets were used to map the transmission of microbial strains and species between adults. Since members of the gut microbiome cannot survive long outside of human hosts, most of them are acquired through physical contact with other humans. This is an image from a review article called Human Microbiome Acquisition and Transmission, and it goes over the different sources of microbiome strains. So, acquisition starts at birth, with the microbes from the mother colonizing newborns during delivery and breastfeeding. And infants continue to develop the microbiome through contact with the mother, with other members of the same household, or with other caregivers. But there's also evidence that there's microbiome transmission between adults. So, this is an article that's mentioned in the study that we're going to look at today. It was published in Nature Microbiology in 2019. And it shows that the microbiome acquisition does occur in adults through social interaction. So they looked at the familial and the social networks of Fiji Islanders, and they also deep sequenced the gut microbiomes of 287 people living in these rural areas in the Fiji Islands. And they found that people living in the same household had high strain similarities, and they could also accurately predict spousal relationships based on these similarities. So the study that we're going to be looking at, they wanted to expand upon these findings, that the findings that adults can share microbial strains through social interactions, but they wanted to look more into the different kinds of social interactions that someone can have, such as between close friends or acquaintances, people who don't live in the same household, and they looked more into how these individuals are spending time together as well. So why is this research important? So social isolation is associated with several adverse health effects. According to the CDC, it's associated with heart disease and stroke, type 2 diabetes, depression and anxiety, suicidality and self-harm, and earlier death. So this is very similar to the adverse health effects of chronic stress. People who experience loneliness have altered levels of stress hormones, specifically cortisol, and their cortisol levels throughout the day are very similar to someone undergoing chronic stress. So how are these findings relevant to us? According to the US Surgeon General, Americans have been spending more time alone and spending less time on face-to-face social interaction. You can see here different forms of social interaction from 2003 to 2020. Social engagement with family members has gone down, and social engagement with friends has also gone down. And according to that study, social networks have also been getting smaller, and people have fewer close friends than ever. And these trends have been accelerating, especially after the COVID-19 pandemic. And I also thought this research was relevant to us as online students. I know that after I finished my undergrad after I finished undergrad, my in-person social networks shrank considerably. And now, as part of the online program, I'm not really having as much face-to-face interaction as I used to. So research in this area is very important to understanding how our social networks shape us, and in the study that we're going to look at, how they shape our microbiome. We know that our microbiome plays a huge role in our health and our physiology, so I'm interested to know if these patterns of social interaction will reflect in the microbiome, and what sort of implications this may have for our health. And maybe we'll be able to use these findings to build stronger communities. If someone's experiencing loneliness, it's more than just a personal feeling. There are ways that our environment and our society can make us more prone to isolation. Someone may be facing discrimination due to age, gender, sexuality. It could be due to the way that our cities are built, where everything's really far away and you need a car to get anywhere. It could be from how our society is shaped, that prioritizes the nuclear family over all other connections. I'm sure you've heard the saying, it takes a village to raise a child. So these are just a few things to keep in mind going forward in our discussion of this paper. So now I'm going to talk about the methodology of the paper. So the researchers did gut microbiome sequencing of 1,787 adults within 18 isolated villages in Honduras. It's significant that they chose these isolated villages because they live very traditional lifestyles there, and the researchers wanted to control for factors such as diet and antibiotics use that would impact the microbiome. And these villages are located in the highlands of Honduras, so this isn't really mentioned in the study, but I imagine that transport is limited in these very mountainous regions, and there's not a lot of back and forth between the villages and the larger industrialized cities. What they used for sequencing was the Illumina NovaSeq 6000. So we know that this is a next generation shotgun sequencing method. It has high throughput, but a small read length. The read length for the 6000 is 250 base pairs sequenced from both ends, which makes it good for human microbiome analysis. You can get an idea of microbial diversity by sequencing all the genes that are present in a sample. And you don't have to reconstruct the whole genome, you can just sequence the 16S rRNA gene to identify the prokaryotes. They also resequenced the microbiomes of 301 individuals within 4 villages 2 years later. And to identify social networks, they surveyed each of the individuals. So they identified the familial connections, such as spouse, parent, sibling, or child. And they also asked questions like, who are your closest friends? Who do you spend time with? And who do you trust to talk about something personal or private? And for the people who reported spending free time together, they asked how often they did so, whether they shared meals, and how they greeted each other. To identify species and strains present in the samples, they used a reference database, and the same program was also used to identify when strain transmission occurred. So basically, if 2 people shared the same genetically distinct strain, this is consistent with a transmission event, rather than if they had acquired the same strain from the environment, although the researchers were not able to rule out environment completely. So we're going to be going over the first main figure of the article, which shows strain sharing across relationship types. So their first finding was that if a relationship is present between a pair of individuals, those pairs share more species and strains than pairs where no relationship is present. So the y-axis here shows us the strain sharing rate, and the way that they calculate this, they divide the number of shared strains by the number of species present in any 2 samples. And from that, they generate these box and whisker plots to compare the median of each distribution. So the percentages you see here at the top of every box is the median strain sharing rate. To determine the statistical significance, they use the Wilcoxon rank sum test, which is appropriate when you're comparing data sets that come in pairs like this. We're looking at any 2 people that have a familial relationship or any 2 people who are friends, with the null hypothesis that the difference between the 2 medians is 0. So let's look at these first 2 boxes here for an example. So if the medians are very similar, like 13.9 and 13.8, if you subtract them, it's going to be close to 0, so that's not statistically significant. If the medians differ more, then the difference is greater than 0, and the difference is statistically significant. So all of these pairings are statistically significant except for the ones with the bar at the top that says NS. And they also observe the gradient of strain sharing based on the type of relationship. So you have more strain sharing with a spouse than with a mother, more strain sharing with a mother than with a child, and so on and so forth. But all pairs that have relationships present share significantly more strains than pairs that have no relationship. There is some strain sharing between pairs with no relationship who live in the same village. You can see at the end of the graph here, the strain sharing rate is 4%. So the researchers, they attribute this to living in the same environment, or maybe there's just strains that get passed on to everyone. And when it's between pairs with no relationship that live in different villages, it's 2%. So that was figure 1 part A, and for parts B through D, they examined strain sharing for pairs that reported spending free time together. So here they looked at how often that the pair spent free time together, and they used the Kruskal-Wallis test to see if the differences in distribution are statistically significant. So this is a test that's appropriate if there's more than 2 samples, like we see here. The test doesn't actually tell you which one of your samples is significantly greater than the other samples, it just tells you that at least one of them is greater. So afterwards they did the Wilcoxon rank sum test again to do the pairwise comparisons. So we see that the pairs who spend time together every day share significantly more strains than those who spend time a few days a week or a few days a month. And again, the samples with the bar that says NS at the top are not statistically different from each other. So in addition to asking the people who spent time together how often they did so, they also asked how often that they shared meals together. So once again, they used the Kruskal-Wallis test to see if any of the 4 samples here were statistically greater than the others. And then they used the Wilcoxon rank sum test for the pairwise comparisons. And they found that the pairs that shared meals almost every day shared significantly more strains than those who shared meals once a week or less. And there were no statistical differences between those who shared meals once a week or less. It's hard to definitively say that strain sharing was due to transmission between individuals. It's possible that the strain similarities are due to eating the same kind of food together. But later we're going to see how the researchers try to control these confounding variables. So for the people who reported spending free time together, they also asked them, how do you greet each other? Once again, they used the Kruskal-Wallis test to see if any one of the samples that you see here are significantly greater than the others. And they found that there were no statistical differences between each of these greetings. This possibly could be due to the small sample size and because there were several different greeting types. So you see here that the kiss on the cheek was associated with the highest rate of strain sharing, but it wasn't significantly greater than the other greetings. I thought it could be interesting if future research focused more on the relationship between greeting types and strain sharing, just because I feel like the type of greeting can vary a lot based on culture. So in Latin American countries like Honduras, it's probably more common to greet people you know with a hug or a kiss than in cultures where there's less public displays of affection. So I'm interested to know how much these patterns in strain sharing can vary by culture. So this is the last graph in figure 1. I had to turn it 90 degrees so that it would actually take up the slide. But basically it's just showing you how they controlled for the other variables that may affect the microbiome. So during the survey, they asked about these other factors such as water source, medication usage, and diet. And they used a covariate permutation approach to determine that if there was a relationship present between a pair, this was more strongly associated with strain sharing than any of the other variables that they looked at. So it's more likely that strain sharing in a pair was due to microbial transmission via social interaction than if they just shared the same strains because, for example, they have the same diet or they're in the same demographic. So we're done with the first major figure of this paper, but I also wanted to go over some of the supplementary figures because I thought that this was interesting. So the researchers found that mothers have a significantly higher strain sharing rate with their children than fathers do. They used the Wilcoxon rank sum test to compare the distributions of each sample, and the rate of strain sharing was significantly higher, but there was no statistical differences between the groups when they looked at species similarity rather than strain. The researchers attributed this greater strain sharing between mothers and their children to the colonization of the newborn with the mother's microbes during childbirth, but it's also possible that it could be due to societal factors. For example, there may be a cultural expectation for women to participate more in the child rearing than men. So here's another supplementary figure. The researchers determined that women were no more likely to share more strains with their direct connections than men. So if you remember earlier in the presentation, there was another study done in the Fiji Islands that also investigated the microbiome and social networks. And in that study, they did find a significant difference in strain sharing between men and women, with women having a higher strain sharing rate than men. So using the Wilcoxon rank sum test, the researchers observed that there was no significant difference between the strain sharing rate in men and women, Strain sharing rate is the one here, the one in orange on the right, and the black bar at the top just shows the p-value for the test. And they also used other indexes to measure the microbial similarity between the direct connections. So the Bray-Curtis Similarity Index, it's here on the left in green. This basically shows how dissimilar two samples are in species composition based on the number of species that they counted in each sample. So once again, they use the Wilcoxon rank sum test to compare the two distributions. So at the species level, men are actually more likely to be similar to their direct connections than women. According to the researchers, part of this is due to brothers being more similar to each other than sisters were similar to each other. And then the Jackard Similarity Index is here in blue. This is another way of analyzing similarity. They just divide the number of species that they have in common by the number of species that they don't have in common. They used the Wilcoxon rank sum test again, and there was no significant difference between the men and the women, even though for the Bray-Curtis Similarity Index, there was a difference at the species level. So this suggests that there may not be that much of a difference in the actual number of species shared in brothers versus the ones shared in sisters, but that the sisters just have more variability in their species abundance. So overall, the reason why this result is different from the one in the Fiji Island study, this could be due to cultural differences. I just thought it would be interesting to point out again how these cultural differences can be shaping the microbiome. It's possible that there could be different expectations on how close these connections are or how they spend time together just based on their gender. And this is a graph from the extended data section. So the researchers also found that if a tie was reciprocated, meaning that for the relationship to count, both of the people have to nominate each other as the closest friend or as the partner, for example. These reciprocated ties, they showed an increased strain sharing rate for all relationship types except for if they were partners. So the paper, it just says that 2,653 ties were reciprocated and then 3,035 weren't, but I'm not sure what the sizes were for each sample or for each relationship type. I imagine that the partners were more reciprocated than not, so maybe that's why, but they never really elaborate on this point. There also seems to be a lot of outliers here. You see the dots at the top of each box are like way above where the whisker ends for each distribution. So basically here, the horizontal line is where the median is, and then the whisker goes up to the largest data point, but only if it falls within 1.5 times % the range from the median to the upper quartile. So then all the dots above where the whisker ends, they're considered outliers. So yeah, this is just something I wish they went more into. I'm curious to see, how many ties were reciprocated versus non-reciprocated for each relationship type. So here they use the model to predict whether a relationship was present based on strain sharing. They use the mixed effects logistic regression model to do this. So first for graphs B and C, they use the model to predict all social or familial relationships, and to account for the other variables that might affect the microbiome, they made 4 different curves, each one represented by these 4 colors. So the red curve is if it predicted based on the strain sharing rate alone, orange if it predicted based on just the shared demographics like age, sex, wealth, education, and religion, green if it predicted based on the strain sharing rate plus age plus sex, and blue if it predicted based on the strain sharing rate plus all of the shared demographics. So the red curve based on strain sharing rate had a moderately strong performance, and its prediction performance increased when the shared demographics were also added to the model. And for graph C, where it says stable ties, that just means it only counted the relationship if it was reciprocated. So for that one, the prediction performance is very similar. For graphs E and F, instead of trying to predict all relationships, it just predicted the non-kin different house relationships. This one didn't perform as well as the one predicting all relationships. That was because it's easier to predict relationships for the people living in the same house since they share the most strains. Same goes for people who live in the same family. But it was still able to predict better than just random chance. The next graphs are from figure 4, where they wanted to see if there was a link between strain sharing and network position. Network position consists of your direct first-degree connections, as well as your second-degree connections, like the friends of your friends. The y-axis here is the strain sharing rate, and the x-axis is the geodesic distance between any two people. So each interval here is the first-degree, second-degree, third and fourth-degree connections. The line in orange represents the null hypothesis, where network position has no effect on strain sharing. They calculated the null distribution by using a permutation approach with 10,000 random samples, and the blue line shows the strain sharing rate by geodesic distance. We see that the first-degree connections have a higher strain sharing rate than the null distribution, with the median strain sharing rate being 7.95%, and that the second-degree connections also have a higher strain sharing rate than the null at 5.10%. And then from the third-degree connection onwards, it's roughly the same as the null distribution. So for the next part of figure 4, they wanted to know if people who were more socially central have a higher strain similarity to the rest of the village. So the degree centrality is based on the number of connections that a person has, and as the centrality increases, the strain sharing rate with all village members also increases. Here they're using the Pearson correlation to measure the strength of the correlation, and the researchers suggest that if someone is more embedded in a community, they're more likely to be exposed to strains that are spreading within that community, making them more microbially similar to everyone in that community. And here they wanted to find out if a socially central person was less microbially similar to their first-degree connections. So they found that as the degree centrality increases, the strain sharing rate with their direct connections decreases. So even though they have more connections and are more similar to the community as a whole, they're less similar to their first-degree connections. The researchers called this a paradox of popularity, where since they have so many social interactions with everyone, they're not spending as much time with just their direct connections. And once again, they're using the Pearson correlation here to test the strength of the correlation. The researchers set out to ask the question, how can face-to-face social interaction shape the microbiome? They wanted to catalog different types of relationships, so not only familial and spousal relationships, or those between people living in the same household, but also close friends and people who you spend free time with. They found that pairs where a relationship is present have significantly greater strain sharing than pairs with no relation, and that strain sharing increases as the amount of time spent together increases, and that strain sharing also increases as the amount of times you share meals together increases. They were able to use a regression model to predict if a relationship was present based on the amount of strain sharing, and they also found that how embedded you are in your community can also shape your microbiome composition. People tended to share more strains with their closest connections, and the more connections that someone has, the more microbially similar they are to the rest of their community, and then people with more connections also share less strains with the people who are closest to them. One limitation of the study is that we can't say for certain that the transmission of strains was due to social interaction, or if it's due to other shared variables such as diet or just living in the same environment. These results may have implications for public health. The researchers suggest that the spread of the microbiome through social interaction could explain the spread of certain phenotypes among people in a social network, such as depression or obesity. So not only could these conditions be explained by genetics or shared behavior, but also by a shared microbiome. My main takeaway from the study is that the microbiome is something that can affect our health in so many different ways, not only on the individual level, but also on the community level. So if you're watching this, take it as a sign to reach out to someone close to you, because these connections may be more important to our well-being than we realize. These are my 5 discussion questions for the group. First, how can proximity to large cities affect the microbiome? The study was done in isolated villages in Honduras, and it made me wonder if strain sharing patterns would be different in more densely populated urban cities, and whether any transport between the cities and the isolated villages could impact the microbial composition of either setting. 2. How can urban planning affect the microbiome? These villages in Honduras were very remote and located in a mountainous region, so there may be less access to infrastructure like public transportation. This is kind of related to the first question, but let's focus on the population density and more on how the cities are built. So considerations like, can you walk from place to place, or do you need a car just to get anywhere? 3. What does the microbiome of a socially isolated person look like? The study focuses on socially central people, but is the microbiome of a socially isolated person different from the rest of their community? And seeing as how the microbiome can affect our mental health, could it be possible that the microbiome composition is a social determinant of conditions like depression and anxiety in these isolated people? 4. To what extent does strain sharing vary by culture? We've looked at social network studies done in Honduras and the Fiji Islands, and in these smaller villages that have more traditional lifestyles. So how do their microbiomes differ from those of other cultures? 5. How can strain sharing impact immunity? Could greater strain sharing within a community bolster their immunity as a whole and have a protective effect? Or does everyone sharing the same strains have a negative impact on community health?